All right, welcome everyone. And you can introduce yourself and start when you're ready.
Great. Hi, everybody. It's good to see some familiar faces in the crowd. My name is Andrea. I'm a MD PhD student in the neuroscience program at University of Minnesota. My doctoral advisor is Dr. Anna Zilverstand, and I'll be talking about sex and gender differences in substance use disorders, specifically an ongoing systematic review of human neuroimaging research. So I don't have any disclosures. So before I start talking about substance use disorder, I do want to clarify why I'm using this combined term sex gender. So as I'm sure I do not have to convince this crowd that sex and gender are not the same thing. So sex refers specifically to biological attributes of humans and animals. It includes physical features, chromosomes, hormonal assays, physical morphology. This is in contrast to gender, which is a socially constructed roles and behaviors assigned to men or women or gender diverse folks who that varies across place and time. But although they are not the same thing, they are related, and we can see that depicted in the orange figure on the right. The expression of genes leads to translation and synthesis of sex hormones, which throughout development, a variety of phenotypes. And the way that your society interacts with those phenotypes around you in a gendered way can affect social factors or lifestyle factors, for example, nutrition, stress, threat reactivity. And there's evidence to suggest that these social factors then can provide a feedback loop onto a variety of biological features, such as sex hormones, as well as onto the transcriptome itself through epigenetic modifiers. I'll be talking specifically about the brain in this talk. So the brain is, of course, of biological origin because it's an organ, but it's also a social organ. So an adult will have experienced a lifetime of very dynamic interactions between the brain and its gendered social environment. So I'm choosing to use this combined term sex gender to kind of capture that nuance specifically for this talk. Can everybody hear me okay? Yeah, okay. So on to substance use. So the gender gap in alcohol use is narrowing. Historically, men have endorsed more alcohol use than women, but this gap is closing, and in kids, and alcohol use in boys and young men have declined faster than in girls and young women. And we can see that depicted on the figure on the right here. So x-axis is time in years, and y-axis the percent of 12th graders who have reported either drinking in the past month or have been drunk in the past month. Squares are boys and circles are girls. And we can see that in earlier times around the 70s to the 90s, there's this gender gap such that boys reported higher prevalence. But as we get into more recent years, that gender gap is closing. In adults, this narrowing gender gap exists as well and is driven primarily by women. And we can see that in the figure on the right. So this is reports of past year alcohol use as well as past year binge drinking on the right. This is past year prevalence. Overall prevalence is in the bold, men is dotted, and women are dashed. And we can see that across the past 20 years or so, the prevalence of both past year alcohol use and binge drinking has remained relatively steady for men. This was all pre-COVID data. And for women, it's been increasing. So it really seems that this gender gap is driven by increasing rates in women. And although I'm using alcohol as a key drug of example, these increasing trends in women is true across many drug types. So this is some recent data from CDC indicating deaths per 100,000 women across time. And we can see that there's been pretty steady increase in drug overdoses in women across a variety of drug classes. So benzodiazepines, antidepressants, benzodiazepines, opioids, et cetera. This is particularly concerning because of this paradigm called the gender risk severity paradox. So although women tend to drink less or use drugs less than men, the consequences of these behaviors tend to be greater. And this is just one example. So on the y-axis here, we have relative risk of liver cirrhosis. On the x-axis, we have alcohol consumption in drinks per week. And we can see that almost every level of alcohol consumption, women have a greater relative risk of liver cirrhosis relative to men. And although I'm using an example of medical sequelae of alcohol use, this is true in other domains as well, including social stigma and legal implications. So it's important to understand this kind of underlying mechanisms of substance use disorder so we can treat or address this narrowing gender gap. And a substantial amount of evidence over the past few years have suggested that underlying substance use disorders can be described by these three functional domains. And those are negative emotionality on the left, executive function, and approach behavior on the far right. Negative emotionality refers to specifically negative emotions, so anxiety, or neuroticism, or aggression, or lack of agreeableness. Executive function refers to kind of your ability to make decisions or inhibit your own behavior, inhibit impulsive motor behavior. And approach behavior refers to kind of this motivated behavior toward a specific cue. So in this case, the cue would be alcohol. So it's motivated behavior towards seeking and engaging with alcohol. Importantly, these functional domains seem to have implications for treatment response. So in a recent paper from Katie Wickowic's lab, sorry, Katie, at University of New Mexico, they characterized a group of problem drinkers as having either drinking as a form of relief, so to reduce negative emotion, or as a form of reward, so having that kind of social salience. And importantly, they identified two subgroups within their sample, one that was drank specifically for high relief and one that drank for specifically high reward. And they actually found that after treatment with naltrexone, which is an opioid antagonist that's used for alcohol use disorder as well as opioid use disorder, they found that only the group that was high on reward evidenced reduction in drinking behavior after receiving naltrexone relative to placebo. So there does seem to be treatment implications for these functional domains. Importantly, they didn't examine executive functioning, so that's another room for growth to help develop this precision medicine for alcohol use disorder. Furthermore, substantial amount of work over the past few decades have gone into understanding some of the underlying neurobiology of these domains. So this is a very simplified figure that shows three stages of substance use. These include the binge intoxication stage, which can be mapped onto this approach behavior domain, the negative affect or withdrawal stage, which can be mapped onto this negative emotionality stage, and this preoccupation or anticipation of use, which can be mapped onto this executive functioning stage. And this is a very simple graph of kind of the neuroanatomy and neurocircuits that underlie some of these behaviors. But all this to say that there seems to be these kind of specific neurocircuits that drive these behaviors. And this is important because this provides us an opportunity to develop novel treatments or use already existent treatments that may target some of these domains. So for example, I have some examples listed here that can target executive control functioning, some pharmacology that may target this reward processing, as well as some stress regulation in red. And although I've listed kind of pharmacology or neuromodulation methods of treatment, this can also be applied to psychotherapy as well. So perhaps cognitive-based treatments that focus on working memory can be targeted toward this executive control, motivation-based interviewing like MI or motivation enhancement therapy, as well as psychotherapy such as CBT or DBT. These are all kind of targeted treatments that we can use based off of these functional domains. One of the ways that we study the neurobiology of these functional domains is using functional magnetic resonance imaging, fMRI. So for those of you who aren't familiar, fMRI is when you stick a person in an MRI scanner, just like you would see at a hospital. When they're in the scanner, you then ask them to do an activity or a task. When they do that task, the brain regions that underlie that task or help that person do that task are increase their neural activity. And because there's this increase in neural activity, there's this increase in neurovascular coupling. So you need to move more blood through your vasculature to those brain regions in order to support those neurons. We can then read that hemodynamic response as this bold response in order to identify regions that are getting more of this blood. So as an indirect activity indirect measure of neuronal activity. So for example, if you stick me in a scanner and then you give me a task of just opening and closing my eyes, when I open my eyes, my visual cortex, which is shown here on the far right, which is in the occipital lobe, will be more active because my eyes are seeing and interpreting the visual cues around me. When I close my eyes, those will be less active. And then we can examine the difference in neural activity between task open and closed eyes, and to specifically target or identify these brain regions that are underlying that task. When we examine substance use disorders, there are many different tasks that we can use. I'm just going to provide an example of 3 of them. So when we measure executive functioning, one potential task is called the stop signal task, and that is when participants are asked to press a button when they see a go cue, which is shown in the little circle here, and to not press a button, so to refrain from pressing the button when they see a stop cue, that is depicted with the X cue here. And because there are many more go trials than stop trials, we develop this prepotent response, or this predisposition to press go. So our ability to prevent ourselves from pressing go, or to refrain from pressing go, is called the stop inhibition, and is one measure of executive function. One measure of negative emotionality, which I highlight here because it was developed at Yale with Rajita Sinha, is asking participants to relay or kind of tell us a recent stressful event that they experienced, and then we play back that script for them through audio. And I'll just read a couple sentences from this example. So this participant's imagery script said, you're in your car with M, you've decided to break up with her. You glance over at her, she looks surprised. Your stomach is in a knot. You know it's the right thing to break up, but your chest is tight. There's a sinking feeling in your chest. She's quiet, you feel hot all over. As you sit down, tears come to your eyes and you start sobbing. So really kind of evoking this negative emotion that's related to the stressful event. And when we think about measuring approach behavior, one of the most common tasks to use is this cue reactivity task, which is really we just show images of a cue of interest, so in this case, alcohol, to our participants while they're in their scanner, and measure their brain function in response to viewing those images. Importantly, sex and gender differences have yet to be probed in many of these tasks. And I think, and that's a detriment to our ability to be able to identify both neurobiologically and sex and gender informed treatments based off of these functional domains. One example of the way that this has been is growing and has been applied is this growing literature suggesting the use of guanfacine as a way to reduce cocaine and alcohol craving. And this is also out of Rajita Sinha's lab here. So guanfacine is an alpha-2 adrenoreceptor antagonist that's FDA approved for ADHD. And some recent work has shown that use of guanfacine has the sex-specific effects such that it reduces alcohol craving in men, in women, but not men. And although I'm showing alcohol craving here, this is also true for cocaine craving, as well as anxiety and negative emotionality. So this is one example of how understanding some of the neurobiology underlying these domains related to substance use disorder can be informed by sex and gender work. So in order to move forward in the field, we have to know where we're at right now. So we did a systematic review of the literature. We identified 579 records over 3 databases, and through a series of kind of identification and screening methods, we identified 34 papers that assess adults' sex and gender differences in substance use disorders using human neuroimaging. So that's not a lot of papers, but you can see that the count of papers by publication year has increased relatively recently. So just to note, the NIH mandated sex as a biological variable for preclinical studies, for animal studies in 2015. And you can see kind of a slight increase, but not as much as you would think. So a lot of room to grow there. Some very basic study characteristics. So the average sample size of men and women was 23 and 24 respectively. But importantly, the most common sample size was 10 women and 17 men. So women tend to be just underrepresented in these samples, which makes it difficult to interpret some of the results. And these are quite small sample sizes. The most common drugs to study were alcohol, nicotine, and cocaine, notably no opioids. And 3 studies examined differences in the menstrual cycle, and 1 study provided reasoning for reporting sex versus gender. When we look at the domains that have been assessed by task, we can see that executive functioning tasks are the minority of tasks that are used. So only 3 studies used the stop signal executive functioning task. More studies use these negative emotionality tasks. So again, that's one example is just the stress imagery task, and these are depicted in the red hues here. And by far, the majority of studies assess this approach behavior. So using this cue reactivity task, and those are shown in the blue, in the blue hues here. Again, these are just examples of some of the tasks that one can use, but these are the most common. When we look at the papers that look specifically at executive functioning, first of all, there's only 3, so very few, if any, conclusions can be made. But we find from these 3 studies that the thalamus is the most frequently reported region of the brain to have a sex gender difference. So on the x-axis here is the different brain regions, which I won't go into detail on, but you can ask me if you have questions. And the y-axis is just the count of papers. For those of you who like looking at brains, here's the thalamus. It's in the subcortex in the third ventricle. When we examine papers that look specifically at negative emotionality, we find more papers, and there are more regions that report sex gender differences probing negative emotionality, and these can be found in the prefrontal cortex, specifically this region called the orbitofrontal cortex, and the subcortex, and this region called the striatum, as well as this region, the posterior cingulate cortex. And all of these regions have kind of unique roles in value-based decision-making and reward-based decision-making. Finally, when we look at papers specifically examining approach behavior, we find that this is, of course, as I mentioned before, the majority of studies. It seems actually pretty evident that the most common region to report a sex gender difference is this region, large region in the prefrontal cortex, specifically the orbitofrontal cortex, kind of ventromedial prefrontal cortex region. And this, again, is involved in this value-based decision-making processes. So overall, there seems to be very preliminary but suggestive evidence that to indicate sex gender differences in these key regions of addiction neurocircuitry, specifically the orbitofrontal cortex for this approach or binge intoxication stage, the thalamus for this executive control or preoccupation anticipation stage, and the striatum, here called the basal ganglia, indicated in this negative affect withdrawal or negative emotionality stage. So there are a few takeaways that I hope you get from this talk. The first is the number one takeaway I hope is just a call to action. So there are only 34 papers, which is really not a lot of papers. There are only 3 papers that included data on menstrual cycling, which seems pretty appallingly low until you look at it in the context of this historical review that was done from Emily Jacobs' lab at UC Santa Barbara, and they tracked all the brain imaging studies that have been done over the past 30 years or so and indicated how many of them took into account women's health factors in blue, gray. And if you can't see how many of them took into account women's health factors, it's because you have to squint. That's that little black smudge in the bottom is how many imaging papers took into account women's health factors. There's only 2% of papers, and of those 2%, 20% mentioned these factors in order to say that they were not going to take them into account. So really kind of a huge dearth in the literature, especially considering that these factors directly affect 50% of the population and indirectly affect the other 50%. So definitely a huge area of growth in this field. The second takeaway is that there does seem to be preliminary support for these sex gender differences in addiction neurocircuitry, again, in these 3 domains of negative emotionality, executive function, and approach behavior. And I think really calling attention to this fact that executive functioning needs to be studied is also a key piece of this. Finally, or not finally, but third, there were no opioid studies, and I think this is particularly concerning given that the ongoing opioid epidemic absolutely affects women. So this is data from the CDC, and you can see this really incredible increase from 2007 to 2017 in opioid overdoses in women from heroin, synthetic opioids, and prescription opioids. So this is really an area of much needed research. And then finally, I think more nuanced investigation about the effect of sex versus gender versus the interaction needs to be done, especially when we think about the brain as a social organ, which I think should be informed by both gender work and social neuroscience. So in summary, again, I hope this is a call to action. So not a lot of work being done. I hope that folks in the audience who are interested will be motivated to continue to do this. There's some preliminary evidence for sex gender differences in these key regions of addiction or circuitry. We need more work on opioids, opioid use disorder, and then more nuanced work on sex gender and the interaction between the two. So with that, if you are in New Haven, you can come say hi at my poster tomorrow. I like to meet new people, so please don't be shy. And then I just want to acknowledge my lab. So my PI is on the left here, Anna Zilverstan, Dr. Anna Zilverstan. Layla Bukhar is another PhD student in my lab in cognitive neuroscience, who's also co-author on this work. The fetal alcohol spectrum disorder lab at the Masonic Institute for the Developing Brain at Minnesota, as well as the MD PhD program, the GPN program at Minnesota, and my funding is on the right. My references on the left and image sources on the right for your reference as well. With that, I will take any questions. I don't know how much time I have. I do want to, so somebody actually submitted a question like earlier this week on the chat that I want to make sure I answer. So the question was, how does addiction affect the female reproductive cycle? Is it harder for people with substance use disorders to track their cycles to understand whether they have premenstrual mood disorder or PMDD or premenstrual exacerbation of another known mental health disorder? So I'll preface this with saying I'm definitely not an expert in reproductive mental health. So I think hopefully I can refer you to some people who might know a little bit more about that. But I did do a brief literature search, and there doesn't seem to be actually a lot of systematic work on the effect of substance use on menstrual cycle regularity. There does seem to be some evidence from a few years ago that shows that women who are long-term methamphetamine users had irregular cycles, specifically in ovulatory cycles. And this also seemed to be supported by some older research in 2003 that found that women who were either received methadone as methadone maintenance or were using or injection drugs had increased odds of irregular menstrual cycles of over 90 days. So a really interesting question. I don't think it's been systematically studied. The other piece of the question was about PMDD, kind of comorbid PMDD and substance use disorders. So again, I actually don't think that this has been really well studied, which I think is just speaks to the lack of a women's mental health focus in this addiction neuroscience sphere. But there was a paper last year that examined PMDD and substance use and found that actually a larger proportion of women with PMS or PMDD in the addiction treatment program relative to a community. So there seems to be potential explanations for common underlying biology or comorbidity between these two disorders. And also the last piece I wanted to say about that was that addiction doesn't exist in a vacuum. There are many other life factors that can affect menstrual cycling and your ability to track that, such as stress, nutrition, comorbid medical disorders. So all those likely play into this kind of women's health, reproductive health piece as well. Okay, how much time do I have, Angela?
You have 1 more minute. And
I have how much?
1 more minute.
Okay, 1 minute. All right.
There's 2 more questions in Whova, and if you don't get to them now, you can always answer them in Whova, and people can respond as well.
You can also email if you have any questions. I can't. How do I see where the question?
I can read.
Yeah, can you read them?
In your opinion, why do you think there is such scarcity of sex and gender differences and addiction in neuroimaging studies, and how can we improve these numbers?
Yeah, so the classic answers in both clinical research and preclinical research was that it was just too complicated to take into effect ovarian sex hormones and the inter-individual variability between that. Within that, I think that's not a good answer anymore. I think we clearly see that it's necessary and we have the tools to study it. We just have to put the effort and the money behind it. And I think that doing this kind of work in terms of identifying really clearly the huge lack and the implications is one way to address that. Yeah.
And we'll ask Do you know the literature in mice or lower models to examine sex chromosome or hormonal effects that could support?
Yeah, so in terms of preclinical research, I'm most familiar with kind of stress-related substance use, and we find that at least in female, that canonically, at least in female animals, seem to have higher relapse rates relative to male stress-induced relapse rates to different substances relative to males. So there seems to be a pretty important component of this cortisol reactivity interacting with these stress hormones. Sex hormones. So I can talk to whoever asked that question later. You can email me or you can find me tomorrow in person. But yeah, that's a great question.
Well, thank you so much for your talk. And I'd like to mention you are a former alumni of Women's Mental Health Committee. So thank you so much for everything.
Yes, I am. Yeah, thanks. I'm wearing my shirt from last year tomorrow, so you can find me that way.
Thank you so much. Everyone will continue the conference. So see you soon.
Thank you.